Imagine a city, but not your standard bustling metropolis.
Right.
It's like this hostile, incredibly smart rogue settlement to just, set up camp inside your body.
Yeah, which is definitely not the traditional way we look at cancer.
No, not at all. I mean, we usually think of a tumor as just a disorganized mass, like a lump of rogue cells that just lost their instruction manual.
Exactly. But when you really pull back the curtain, it is so much more sinister than that.
It's actively hacking into your body's power grid. It's rewriting your internal internet protocols, and it's even turning your own homeland security forces into its personal bodyguards.
It forces us to completely abandon that old view. I mean, for decades, oncology looked at tumors in isolation, like they existed in a vacuum.
Right, just a localized problem.
Exactly. But this rogue city is deeply plugged in. It is actively tapping into the very neurological and metabolic wires that keep you alive, and it manipulates them to serve its own survival.
And for this deep dive, our mission is to unpack the exact mechanics of that manipulation. We're pulling apart a really dense, fascinating 2026 review paper. It's called Neuroimmunometabolic Coevolution in the Tumor, Micro, and Macroenvironment.
It is a mouthful, but it's a brilliant paper.
It really is. And our goal today is to map out for you, the listener, exactly how cancer hijacks three distinct systems all at once: the nervous system, the immune system, and the metabolic system.
And it does this locally, right, where the tumor is, which we call the tumor microenvironment, or TME, but also systemically across your entire body, which is the tumor macroenvironment.
So to really grasp this, we have to start with the core concept governing this entire paper, the neuroimmunometabolic triangle.
Right. It's this continuous three-way crosstalk. Basically, nerves release chemical signals.
Okay.
And those chemicals physically alter how nearby cells burn energy. That's their metabolism.
And then that altered metabolic state dictates how your immune cells behave.
Exactly. And cancer just sits right in the middle of this triangle, pulling all the strings.
So let's use a visual mnemonic to keep this straight for you as we go through. Picture the peripheral nervous system as three vulnerable infrastructure networks. First, you have the power grid. That's your sympathetic nervous system, your fight-or-flight response.
Right.
Second, the cooling system, your parasympathetic nervous system, responsible for rest and digest. And third, the alarm system, your sensory nerves that feel pain.
I love that breakdown. It makes it so much easier to track.
Yeah, so let's start by looking at how a tumor hacks that first network.
Yeah.
How exactly does it hijack the power grid?
Well, the sympathetic nervous system, it originates in the thoracolumbar region, so basically the middle and lower back area of your spinal cord.
Okay.
And it sends microscopic fibers out to practically every organ. Its primary chemical messengers are norepinephrine and epinephrine.
Which most people probably just know as noradrenaline and adrenaline.
Exactly. And these chemicals bind to specific docking stations on cells, which are known as alpha and beta adrenergic receptors. Normally, if you encounter a threat, this system just floods your body with adrenaline, diverting energy to your muscles so you can fight or run.
But the tumor wants that energy for itself. And the paper details this incredible mechanism where sympathetic neurons actually co-develop alongside blood vessels right inside the tumor.
Yeah, that part is wild.
Right. The nerves release norepinephrine, which then locks onto a specific docking station, the beta-2 adrenergic receptor, on the cells lining those blood vessels.
And that triggers a metabolic shift called aerobic glycolysis.
Which is, it's a highly inefficient but incredibly fast way to burn sugar, right?
It is. It's essentially paying the biological construction workers in pure sugar and energy drinks just to lay pipe faster.
That is exactly what it is. It forces those cells to rapidly build the tumor's blood supply. That process is called angiogenesis. The tumor is literally forcing your nerves to build its own plumbing.
But it doesn't stop with the plumbing. I mean, those same stress hormones also hit beta-2 receptors directly on the cancer cells themselves. We see this really strongly in gastric and colorectal cancers.
So what happens when norepinephrine hits the cancer cell directly?
When it hits that beta-2 receptor, it flips a genetic switchboard inside the cancer cell. It's known as the pKa-CREB1 pathway.
Okay, I always picture pathways like that as a row of dominoes. Once that receptor falls, what is the final domino in that pKa-CREB1 line doing?
The final domino is the mass production of enzymes specifically designed for glycolysis. It completely rewires the cancer cell's internal factory to consume massive, gluttonous amounts of glucose.
So it's gorges on sugar.
Yeah, which gives the tumor the rapid energy it needs to aggressively divide. And honestly, it also highlights a pretty harsh reality about chronic stress.
Right, because if someone is under chronic psychological stress, their body is constantly flooded with these exact hormones.
Exactly, which means chronic stress can actively fuel tumor progression by keeping that exact metabolic pathway turned on all the time.
Wow. But the metabolic hacking goes even further. There was this detail in the paper that just felt straight out of science fiction to me. In breast cancer models, peripheral neurons don't just send chemical signals across a gap.
No, they physically reach out.
Right. They form tiny connecting tubes called nanotubes, and they transfer their own functional mitochondria directly into the breast cancer cells.
It's a truly staggering biological maneuver. I mean, mitochondria are the powerhouses of the cell. They handle oxidative phosphorylation, which is the high-efficiency engine for making ATP energy.
So by donating their own mitochondria, these neurons are directly giving the tumor extra batteries.
Exactly. And this massive boost in ATP, along with improved redox balance, it's a major driver of metastasis. The nerves are physically arming the cancer to spread.
Unbelievable. But while the tumor is gorging on all this stolen energy, it also has to worry about the cops, your immune system. And this is where the divide between the beta receptors and the alpha receptors becomes really critical.
Right. So the overarching rule in this microenvironment is that beta adrenergic signaling suppresses your anti-tumor immunity,
Okay.
while alpha adrenergic signaling actually promotes it.
So beta is the bad guy for immunity. Alpha is the good guy.
Broadly speaking, yes. So consider your CD8 positive T cells. These are the specialized killer cells designed to hunt down and destroy cancer.
Right.
Well, they are absolutely covered in beta-1 receptor.
So when the tumor's sympathetic nerves pump out norepinephrine, it hits those beta-1 receptors and triggers what the paper calls T-cell exhaustion.
Exhaustion in a very literal cellular sense. The T cells just stop dividing. They stop releasing defensive cytokines, which are, the chemical weapons they use to destroy threats.
And perversely, activating that receptor changes their navigation programming too, right? It makes them migrate directly toward the sympathetic nerves that are neutralizing them.
Yeah, it's like a tractor beam pulling the security guards right into a trap. Meanwhile, activating alpha-2 receptors on macrophages, which are the immune system's Pac-Men, that can actually trigger a robust attack against the tumor.
But the tumor specifically cultivates the beta receptor pathway just to shut the whole response down.
Exactly. And we know this causality is real because researchers can actually reverse it in the lab.
How do they do that?
They use a chemical tool called 6-OHDA, which selectively destroys sympathetic nerve endings. So when they basically cut the power grid, the tumor's growth slows.
Wow.
And in even more advanced models, scientists have engineered CAR-T cells to secrete antibodies against nerve growth factor, or NGF.
Oh, so by neutralizing the growth factor, they starve the sympathetic nerves.
Right. It effectively cuts the tumor's power lines and restores the T cell's ability to fight back.
Okay, so let's do a quick 5-minute milestone recap here. If I'm putting all these pieces together for you, the listener. Cancer is a systemic disease manipulating this triangle. It uses your sympathetic fight-or-flight nerves to force blood vessel construction. It supercharges its own sugar consumption, and it completely exhausts your killer T cells using those beta receptors.
That's the power grid hack in a nutshell.
So here is a question for you to ponder. If chronic stress floods your body with epinephrine that hits those beta receptors and suppresses your immune system, how might managing your daily stress literally change your cellular defense against disease?
It really bridges the gap between our psychological state and the physical microenvironment of our cells.
Which transitions us perfectly to the second vulnerable infrastructure network, the cooling system, the parasympathetic nervous system, or PNS. If the sympathetic system is fight or flight, this is rest and digest.
Exactly. And the PNS largely originates high up in the brainstem in specific nuclei, like the dorsal motor nucleus of the vagus, or DMV.
Okay, and its primary messenger isn't norepinephrine, right?
No, it's acetylcholine, which binds to muscarinic and nicotinic receptors. In a healthy body, this system's job is just to put the brakes on, to calm things down.
But a tumor is a master of exploiting that calm. The paper outlines a striking metabolic example involving lung cancer. I mean, we all know smoking is linked to lung cancer, but the mechanism is deeply neurological.
It is. Nicotine physically binds to nicotinic receptors on the surface of non-small cell lung cancer cells.
And that binding event causes the cancer cell to upregulate a protein called HIF-1 alpha.
Right, and HIF-1 alpha is essentially a panic button for oxygen deprivation.
So when it's activated, the cell acts as though it's suffocating.
Exactly, which drastically enhances the Warburg effect. That's the cancer's preference for burning sugar, even when there's plenty of oxygen available.
And the paper notes this signaling alters the intracellular pH too. The inside of the cell changes its acidity, which physically alters the cellular scaffolding, making the cell highly mobile and driving migration.
So the cooling system is actually manipulated to make the cancer move faster.
Right, but how does it use this system to shut down our immune defenses?
Through this really elegant biological mechanism called the cholinergic anti-inflammatory pathway.
Okay, break that down for us.
Normally, the vagus nerve releases acetylcholine to prevent your immune system from overreacting to an infection. Because if an immune response goes too far, it can cause deadly tissue damage or sepsis.
So the vagus nerve prevents this by commanding a ceasefire.
Exactly. It binds acetylcholine to the alpha-7 nicotinic receptor on macrophages, commanding them to stop releasing inflammatory cytokines like TNF and IL-6.
It forces a ceasefire to protect your organs. But inside the tumor microenvironment, that ceasefire is deadly. The tumor wants those macrophages sedated.
Yeah, and those sedated macrophages are now called alpha-7 positive tumor-associated macrophages, or TAMs.
TAMs.
Right. They transform from hunters into highly immunosuppressive security guards, basically just standing around the tumor and protecting it from the rest of your immune system.
And the manipulation extends to your B cells too, right? The factories that produce antibodies.
Yes, chronic stimulation of the vagus nerve causes a specific subset of your own T cells in the spleen to release acetylcholine.
Wow, your own T cells release it.
Yeah. And that acetylcholine then hits receptors directly on the B cells, severely suppressing their germinal center responses.
I always picture the germinal center like a military training academy for B cells. It's where they learn to produce high-affinity, perfectly tailored antibodies.
That's a great analogy.
So by flooding the academy with acetylcholine, the parasympathetic system forces all the cadets to go to sleep. It entirely neutralizes the antibody arm of your immune response.
So if we synthesize this cooling system hack, the tumor is exploiting the parasympathetic system's natural ability to calm inflammation, using acetylcholine to sedate your macrophages and shut down your B cell factories.
Think about the implications of this for a second. Knowing that your own T cells can synthesize their own acetylcholine, how do you think our own immune cells might accidentally be contributing to this tumor-protecting calm?
It is biological judo. I mean, the tumor just uses our own protective weight against us.
Wow. Okay, let's move to the third sabotage network, the alarm system, the sensory neurons.
These are the nociceptive or pain-sensing neurons.
Yeah.
They're characterized by receptors like TRPV1.
Okay.
But the crucial anatomical quirk of sensory neurons is that they are pseudo-unipolar.
Okay, let's translate that out of the textbook. I'm assuming pseudo means it's not acting like a normal one-way street.
Precisely. We typically think of sensory nerves as input-only cables, right? They detect a burn or a cut and carry that signal away from the tissue up to the brain so you feel pain.
Right.
But because they are pseudo-unipolar, the traffic goes both ways. They are also efferent, meaning they can release highly potent signaling chemicals right back into the tissue they are monitoring.
And this two-way street enables an incredible survival tactic. The paper describes oral cancer growing in a nutrient-poor, low-glucose environment. The tumor is quite literally starving.
So what does it do?
To survive, the cancer cells secrete nerve growth factor, NGF. This acts as a beacon, recruiting the local sensory nerves. And then those nerves release a peptide into the tumor called CGRP, calcitonin gene-related peptide.
And that CGRP then binds to the cancer cells and triggers a process called protective autophagy.
Right, and autophagy literally means self-eating.
Exactly. The cancer cell begins to break down and consume its own damaged internal components, recycling them just to survive the starvation. The pain nerve is functioning as a life support system.
And the researchers actually visualized a separate version of this feeding process in the lab, which is fascinating. They built these microscopic, microfluidic devices separating the main bodies of sensory nerves from pancreatic cancer cells.
And the only connection between the two chambers was the long microscopic axons of the nerves, right?
Exactly. And they watched these sensory axons physically drop off the amino acid serine directly into the cancer's environment, providing the exact nutritional building blocks the tumor needed to grow.
The alarm system basically acting as a localized grocery delivery service.
Unreal.
And on the immune front, that same peptide, CGRP, acts directly on CD8 positive killer T cells, triggering a cascade that forces them into that exact state of exhaustion we discussed earlier.
So they're feeding the tumor and exhausting the T cells.
Yes. In animal models, when surgeons physically sever these sensory nerves, essentially performing a sensory denervation, the microenvironment radically shifts.
What happened?
The number of active killer T cells skyrockets, while the number of regulatory T cells, which normally suppress immunity, plummets.
Which brings us to a phenomenon the paper highlights that absolutely blew my mind. The long-distance hack.
Oh, this is incredible.
When oral cancer is under attack by the immune system, it activates local sensory nerves.
Yeah.
And those nerves act like a fiber optic cable, bypassing local defenses and sending a signal all the way to the tumor-draining lymph nodes.
The lymph nodes are essentially the command outposts for your immune system. So when that sensory signal arrives, it suppresses the entire node. The lymph node drastically reduces its production of a chemical messenger called CCL5. And because CCL5 levels drop, the macrophages back at the tumor site lose their aggressive signaling. They polarize right back into a pro-tumor immunosuppressive state.
So the tumor felt the heat, sent a distress signal through the pain wires to the lymph node, and the lymph node remotely turned off the immune response.
It is brilliant, terrifying engineering.
So at the 10-minute mark here, let's do a quick recap of the last two systems.
Sure. So 1, parasympathetic nerves use acetylcholine to suppress inflammation, which unfortunately protects tumors. 2, sensory nerves act as a two-way street, both feeding tumors amino acids like serine and acting as a long-distance communication wire to corrupt lymph nodes.
Right.
And 3, the chemicals involved here, like CGRP and nerve growth factor, are turning out to be key targets for future therapies.
So the question for you, if immune cells fighting the tumor actually stimulate these sensory nerves to grow, which causes cancer pain, could treating the pain with nerve blockers also help kill the tumor?
That is the exact paradigm shift happening in oncology right now. Treating the pain might be a direct therapeutic intervention against the disease itself.
Wow. Now we have to zoom out. Because we've mapped the local neighborhood, the TMaE, but the tumor's ultimate prize is the systemic network, the tumor macroenvironment. It wants access to the body's central command, the brain.
Because the brain is the master regulator of both metabolism and immunity, cancer exploits long-distance circulating factors to basically rewrite the brain's software.
And the most devastating consequence of this is cancer-associated cachexia, or CAC. Cachexia is that severe muscle wasting and catastrophic weight loss seen in advanced cancer patients.
And to be clear, this isn't just a patient losing their appetite, right? The body is actively dismantling its own muscle and fat.
Right. The paper frames this as a vicious inflammation metabolic disorder loop. Let's walk through this loop slowly. How does the tumor actually initiate this systemic breakdown?
It begins at the peripheral tumor, which secretes a long-distance messenger protein called CCL2.
Okay, CCL2.
That CCL2 travels through the bloodstream and induces neuroinflammation in a very specific part of the brainstem, the DMV, which we noted earlier as the origin of the vagus nerve.
Okay, so this central inflammation essentially hot wires the vagus nerve, sending a corrupted signal down to the liver.
The brain is basically sending a malware update to the liver's manufacturing plant.
Exactly. And the vagus signal forces the liver to drastically drop its levels of a regulatory protein called HNF4 alpha.
Right. And plunging HNF4 alpha levels trigger massive metabolic reprogramming in the liver, unleashing a huge spike in a different protein called LCN2.
And LCN2 is a cachectic driver.
Oh, absolutely. It floods the bloodstream, crosses the blood-brain barrier, and hits the brain again. Its primary effect is to completely kill the host's appetite while simultaneously aggravating systemic inflammation.
But a smart city has redundancies. The tumor isn't just relying on that one CCL2 loop, is it?
No, it also releases a neuropeptide called GDF15. This travels directly to the brainstem, locking onto specific receptors, GFRAL-expressing neurons, in brain regions called the AP and NTS.
And what's the purpose of hitting those neurons?
The sole biological purpose is to further obliterate the appetite. The tumor is systematically starving the host to free up structural nutrients for itself.
It is systemic parasitism on a grand scale.
Yeah.
But the brain's regulation goes beyond just metabolism. We have to look at how the tumor hacks the brain's control over the immune system, specifically focusing on the hypothalamus, and a region called the paraventricular nucleus, or PVN.
The PVN is essentially the war room for brain-body immune communication.
And it has a dual nature, right?
It does. If we stimulate specific neurons in the PVM, like those that release beta endorphins or oxytocin, it sends a massive boost to the immune system. It ramps up natural killer cells and CD8 positive T cells, which actively hunt and suppress cancer.
But the tumor aims for the other side of the room. It sends signals to activate CRH neurons in the PVN. And when those fire, they trigger the HPA axis, the hypothalamic pituitary adrenal axis.
Right.
And when the adrenal gland gets that signal, it floods the body with glucocorticoids like cortisol, pure stress hormones.
And tying it all back to the power grid from earlier, we know what stress hormones do. They are profoundly immunosuppressive. They hit those beta receptors, exhaust the T cells, and empower the protective macrophages.
So the tumor literally induces a state of systemic neurological stress to shut down the body's defenses.
Exactly.
Now, the most startling part of this brain-immune connection involves how the brain stores data. The paper discusses the insular cortex, or the IC. I literally had to read this section twice. The brain actually records memories of immune responses.
It really does. The insular cortex encodes and retrieves specific immune memories. When your body goes through an inflammatory event, the IC records the exact pattern of neuronal activation.
Okay.
And in laboratory settings, researchers discovered that if they artificially reactivate that exact specific neuronal ensemble in the IC, the brain will instantly recreate that physical inflammatory state in the body.
Even if the original pathogen is gone.
Even if the trigger is entirely gone.
That is insane. So summarizing this systemic hack, tumors release chemical messengers that travel to the brain, rewiring the liver to cause severe muscle wasting, obliterating the appetite, and manipulating the hypothalamus to flood the body with immunosuppressive stress hormones.
That's the macroenvironment in action.
But thinking about that insular cortex, if the brain can remember immune responses, could we one day train the brain to neurologically recall and trigger a massive targeted anti-cancer immune response simply on command?
While it sounds like pure science fiction today, the fundamental neural architecture to execute that kind of internal command already exists inside us.
Which brings us to the ultimate question. We've mapped the entire grid. Cancer is not a localized lump. It is a highly connected systemic disease. It co-opts the sympathetic power grid to burn sugar and build blood vessels.
Yeah.
It hijacks the parasympathetic cooling system to sedate your immune forces. It manipulates the sensory alarm system to feed itself and corrupt your lymph nodes. And it hacks the brain to drain your body's energy. How is any of this clinically actionable? Like, how do we disconnect the city?
Well, the inherent advantage here is that because cancer is using our existing hardware, we already possess tools to disrupt it. We can utilize physical interventions like targeted radiofrequency ablation to essentially burn and sever the specific nerve cables feeding the tumor microenvironment.
But we also have an entire pharmacy of existing drugs we can repurpose to cut the communications, right?
Absolutely. Beta blockers, which millions of people take daily for high blood pressure or heart arrhythmias, they can be deployed to block the sympathetic nervous system from exhausting T cells.
And what about the sensory nerves?
CGRP antagonists and substance P antagonists, which are medications currently prescribed to treat severe migraines, they can be repurposed to stop sensory neurons from delivering nutrients and suppressing the immune response.
So the future of oncology isn't just going to be about targeting the mutated DNA inside the cancer cell. It's moving toward personalized neuroanatomical treatment.
Exactly. A doctor will look at where the tumor is, map the specific neural networks plugging into it, and chemically or physically cut the power.
It demands a completely integrated view of human biology. We have to treat the whole ecosystem, not just the rogue cells.
We really do.
To leave you with one final provocative thought from the future perspectives of this paper, we just spent a lot of time discussing how peripheral tumors release factors that travel up to the brain to modulate neural circuits, causing cachexia and immunosuppression.
Meaning the brain is actively receiving signals and reacting to the tumor's presence.
Right. So the brain is likely sensing the tumor long before the first physical symptoms appear. Long before a patient feels a lump or develops a cough, the brain's electrical and chemical activity shifts the moment the tumor starts broadcasting those SOS signals.
It has to, mechanically speaking.
So what if, in the near future, the earliest screening for cancer isn't a blood test looking for biomarkers, and it isn't an MRI scan of your organs? What if the very first diagnostic tool is a simple, non-invasive monitor of your brain activity?
That is an amazing thought.
If your brain knows the tumor is there before you do, how do we finally learn to listen to it?
It would fundamentally alter the timeline and the methodology of diagnosing disease.
It changes everything. The next time you think about your body, remember, it's not just a collection of isolated parts or individual organs. It is a highly connected, constantly communicating smart city. And keeping it safe means understanding and protecting the grid. Thanks for taking this deep dive with us. Keep questioning, keep learning, and we'll see you next time.